Not long ago, a young child was admitted to a hospital with a sudden high fever. Basic tests were done and everything looked normal. Yet the child kept getting weaker. The doctors knew something was wrong, but the usual biochemical test couldn't explain it. So they tried something different. They didn't look at the child's DNA, they looked at the RNA inside the cells and changed everything. They discovered that a set of antiviral genes had suddenly switched on, a clear sign that a hidden viral infection had begun, long before symptoms became visible. The transcriptome had already recorded the danger. This is the power of transcriptomics. It reveals biological changes before the disease appears, and this makes it one of the most powerful tools in modern biochemistry. Hello everyone, I'm Supranto from Biotechnology program, BRAC University. We are going to talk about transcriptomics in biochemistry. First of all, what is transcriptomics? If DNA is the full library of life, transcriptomics tells us which page the cell is reading right now. It studies all RNA molecules, the transcriptome, to understand which genes are active, which are silent, and how biochemical pathways are shifting in real time. Even the word explains itself by transcript and omics. Transcript means RNA copied from DNA and omics indicates studying everything together. So, transcriptomics means studying all RNA transcripts at once, like listening to the live broadcast of a cell. How does it work? Biochemists extract RNA, a fragile molecule that degrades quickly, convert it into cDNA, and then analyze it using microarrays. Detect thousands of known gene expressions, then RNA sequencing reads every RNA molecule, even rare ones, then single-cell RNA sequencing studies each cell individually. These tools show us which genes turn on during stress, which shut down during treatment, and how biochemical pathways react inside the cell. Why is it important in biochemistry? Because RNA controls biochemical reaction. A gene turns on, then RNA is made, then DNA makes a protein, and proteins run metabolism, immunity, signaling, and everything. That is why transcriptomics helps us understand early markers of gene, how immune cells respond, why patients react differently to drugs, and how metabolic diseases like diabetes begin at the molecular level, and lastly, how microbes or plants respond to stress. It's used in medical research, biotechnology and drug development, microbial engineering, and crop improvement. Transcriptomics doesn't show us a blueprint, it shows us a biochemistry in reaction. Whether it's a patient, a plant, a microbe, or a single cell, the real story is happening deep inside, at the molecular level. Transcriptomics lets us read that story, the whisper of genes, the struggles of cells, the shifting biochemical pathways that define health and diseases. It shows life not as a still image, but as a dynamic movie happening right now. That is why transcriptomics in biochemistry is transforming modern science, because it finally lets us see life in motion. Thank you so much for staying with us.